Sebastian. In this video, we are going to discuss about translocation of proteins to peroxisomes. Peroxisomes are a different kind of cell organelles when we compare peroxisomes with mitochondria and chloroplast. That is because peroxisome, you know that it has a single membrane, single bilayer, not like chloroplast and mitochondria. And also the peroxisomes have no genome of its own and it does not synthesize the proteins. Therefore, peroxisomes are slightly different from chloroplast and mitochondria. We are going to look at how proteins are translocated to peroxisomes. Let us see that all the proteins meant for peroxisomes are synthesized in the cytoplasm and they have to be transported. If you look at the origin of peroxisomes, we know that they have originated from endoplasmic reticulum and they are found in eukaryotic system. Maybe they have evolved when photosynthetic bacteria evolved. With the photosynthesis, what happens is the oxygen level increases. We are aware that oxygen is highly reactive and this oxygen must be contained. In order to contain this oxygen, probably peroxisome must have evolved. And you will see that the peroxisome has many enzymes like catalases, etc., which or urate oxidase. They use oxygen for several reactions, especially catalase. Now, when mitochondria evolved, they would have adopted all the reactions of the peroxisomes and clubbed it with the production of ATP. That is how probably mitochondria would have evolved. Whereas this ATP producing facility is not present in the peroxisomes. Let us try to, first of all, look at some of the functions of peroxisomes, which is very interesting. Peroxisomes primarily are used for removing hydrogen from a substrate molecule by using oxygen. Let us assume this is a substrate molecule. If the peroxisomal enzymes can remove by using oxygen, it is capable of withdrawing this hydrogen. So you will have an H2O2 plus a substrate molecule. This is one of the important reactions that is carried out by peroxisomes. Now, as you know, H2O2 is also a harmful product in a cell and this has to be eliminated. How is it eliminated? So the enzyme catalase will act. Catalase will convert H2O2 into H2O plus O2. 2H2O into 2O2. So catalase will convert and make that into harmless water molecule. These are some of the functions of peroxisome. Other important functions of peroxisomes include, especially in plants and in yeast, peroxisomes are the site where beta oxidation occurs. So you are aware that beta oxidation will convert a fatty acid into acetyl-CoA. And now this acetyl-CoA will be transported into cytoplasm and it is used for many biosynthetic reactions. Peroxisome is also the location for the first step of plasmalogen metabolism. So plasmalogen is so important for the formation of myelin sheath. So this first step is taking place in the peroxisomes. Peroxisomes are also important for photorespiration. Another important function. And it is important for in the germinating seeds. It will convert fatty acids into sugar molecules, which is needed in germinating seeds by a pathway known as glyoxylate pathway. You can see how many important functions this peroxisome is carrying out. Very interesting aspect about peroxisome is it is a highly adaptive organelle. Adaptive because you allow a yeast cell to grow in a medium containing methanol. So the peroxisome will grow to that level, it will have high content of methanol degrading enzymes. Allow yeast to grow in a medium containing fatty acid. It will synthesize a lot more of enzymes which can degrade fatty acids, and this will be present in the peroxisome. So peroxisome is a kind of it's an organelle which can adapt to an environment. We are able to see the varieties of function this small molecule that is carrying out. Let us also look at how exactly these peroxisomes are formed. As you know, they are formed from endoplasmic reticulum. So if you consider this is an endoplasmic reticulum, they are formed by budding. So a small bud is formed, and this bud is going to be the peroxisome. Now remember, this peroxisome must have, as it is budding, it must have the required proteins, especially the translocating proteins present on it. So these translocating proteins are also present on this membrane as it is slowly getting bud off. Now this will form a structure like this. So it is forming a bud, and this is facilitated by an important protein called PEX-19 is a protein. There are two other PEX proteins. One is PEX-3, another one is PEX-15. This protein also helps in forming this complex, assembling the required proteins in the structure of the newly formed peroxisome. Now what will happen is, now this peroxisome, it will have certain proteins that is coming from the endoplasmic reticulum, and other proteins, okay, they are incorporated into this structure. As it is growing, the peroxisome will incorporate in its structure. So over a period of time, you will see this is having many proteins, and these are the translocators, and this will grow. And it will grow slowly, it will grow to another peroxisome, and with the structures and the protein inside distributed, and slowly fission occurs, and they converted themselves into two peroxisome molecules. So this is how new peroxisomes are formed in a cell. So they are formed from the endoplasmic reticulum through budding and forming a vesicle, incorporating proteins from the cytoplasm. Then they enlarge, again divide themselves into another peroxisome, and that is how multiplication of peroxisomes occurs. Having understood the structure and the functions of peroxisomes, let us look at how proteins which are synthesized in the cytoplasm are translocated into the peroxisomes. Now, in the proteins that has to go to peroxisomes has got a very specific sequence, and this sequence is, we call it either PTS. PTS stands for peroxisomal target sequences. So let us call one of these PTS as PTS1. We are aware of the structure of PTS1. It is a 3 amino acid sequence, that is serine, lysine and leucine. Serine, lysine and leucine, it is called SKL sequences. Now, this SKL sequence, which is denoted by PTS1, will direct the protein to the peroxisome. Now, let us denote, remember, and also remember that the protein that is going to the peroxisomes are folded proteins. They have carried out experiments, scientists have carried out experiments in order to check this particular fact. We are aware that most of the soluble proteins soluble proteins in the cytoplasm will have a size of about 9 nanometer. Now, they have attached to a protein a gold particle in the size of 9 nanometer. They have attached. And it was observed that if there is this PTS1 signal, this protein is getting assembled, getting incorporated into the peroxisome. That is how they proved that up to 9 nanometer protein in the folded form, this protein is capable of directly getting into the peroxisome. A very simple experiment that is very revealing, which is different from other proteins which has to be unfolded, whereas this protein in peroxisome, it can be the folded protein can be translocated. Let us look at a folded protein, and that has got the PTS signal. So this is the PTS signal 1, and this protein is to be get inserted into the peroxisome. Now there is a receptor, a soluble receptor present, and that soluble receptor, we shall denote it like this. This soluble receptor is called PEX5. So PEX5 is a soluble receptor which is capable of identifying the signal. Now this will identify the signal and this will bind. If the signal binds and the protein is here, the folded protein is there, it is bound to PEX. Now this has to be translocated. Now translocation is, this is a peroxisomal membrane, and this is a cytosol, this is a peroxisomal membrane, and this is the lumen or the matrix of the peroxisome. Peroxisomal matrix. Now there is a translocating protein, and this translocating protein is it's a complex formed with 3 PEX proteins, and these 3 PEX proteins, so let us put it this way, these are the 3 PEX proteins, and imagine this also side also there will be PEX protein. And this particular unit, which will act as a translocon, is assembled with the help of PEX 13, 14 and 17. 13, 14, 17 PEX will assemble and it forms a kind of a channel. Now in this channel, this PEX 5, 2 of them will join together. These are the PEX 2 join together, and this is with the signal inside, and here is a protein that is bound, the protein that is bound. Now this is, now it is get translocated into the peroxisome. In the matrix of the peroxisome, it is released. The protein is released in the peroxisome. So now once the protein is released, now this is empty. If the PEX 5 is empty, now the PEX has to be separated from this PEX 13, 14, 17 complex. How to separate this PEX 5? Now in order to separate PEX 5, there is another membrane bound complex. Now this membrane bound complex is again, it is another 3 PEX proteins. These are PEX 2, PEX 10 and PEX 12. That forms a complex. Now this complex, what is the function of this complex? The function of this complex is to attach a ubiquitin molecule. So let us clear this portion. It is to attach a ubiquitin molecule to this PEX 5. It will attach 2 ubiquitin molecules to PEX 5. Attaching a ubiquitin molecule to PEX 5 is an indication that this has to be separated from this particular complex. The PEX 5 must be separated. Now that separation and release of the PEX protein, this is PEX 5, release of that is mediated by 2 other PEX proteins. One is PEX 1, other one is PEX 6. So these 2 proteins will mediate the release of the PEX 5 from this particular complex. That requires hydrolysis of ATP. ATP is hydrolyzed to ADP plus PI. That energy is utilized for separating the PEX 5. Once the PEX 5 is free and it is in the cytoplasm, the same cycle will repeat. It will bind to a protein with the SKL signal. Again, it will bind to this particular complex, transfer the protein inside. Then again, PEX 21012 complex will add a ubiquitin, separate it, and this cycle continues. This is how proteins are translocated to the peroxisomes.